In this video we will discuss about Tscm CAR-T failure insights. So basically the result shows about the deep insight of the patients who does not respond to this Tscm based CAR-T cells. So they will start with by comparing the peak values of CAR-T cells in the standard CAR-T cells and the Tscm based CAR-T cells between responder and non-responder. So for the standard CAR-T cells treated cohort, they see that there is a significant difference between the peak value of the CAR-T cells in the responder compared to the non-responder. However, when they check it for the Tscm based CAR-T cells, they do not see any significant difference of the CAR-T cells reaches to its peak value in the responder and non-responder. Afterwards, they check the patients individually. They investigate the patients individually who does not show a response towards this Tscm based CAR-T cells. So they started with the patient number 23. So in this case, the good thing is that the CAR-T cells expansion occurs. However, they did not show any. They did not show tumor suppressions, or the tumors grow uninterruptedly in the bone marrow and blood. So the panel B shows the CAR-T cells expansion. It is around between 10 to 12 days, which is close to the end of second week. And also the expansions markers are high. That is Ki67 markers are high in this CAR-P positive cells, indicating that they are expand and proliferate in the patient number 23. However, when they check the cancerous cells in the three different places, that is in the bone marrow, blood and B cells, you can see that in the B cells the cancerous cells goes down, whereas in the case of bone marrow and blood the cancerous cells goes up almost uninterruptedly. Afterwards, they did the further analysis in terms of the CD19 expressions. So before this CAR-T cell treatment, when they check this B all cells, pre-B cells, which is also the cancerous cells, and the all B cells, and when they compare between the pre and post CAR-T cells CD19 expressions, they are able to find out that in pre-B cells, which is the immature cancerous cells, cancer precursor cells, there is a CD19, there is an enrichment of low CD19 expressed pre-B cells. That is the signal goes mean fluorescence intensity goes down from 1376 to 1264, indicating that these precancerous B cells that has low CD19 expressions are not being able to react or detect by the Tscm based CAR-T cells, which is also in line with the literature that these CAR-T cells works efficiently on the high CD19 antigen expressions target cells. So, and afterwards in the patient 23, the reason could be that the low density CD19 expressions in the bone marrow and blood cells, peripheral bone marrow and peripheral blood cells, that could be the reason that this Tscm based CAR-T cells does not act on the cancer cells present in bone marrow and blood. Afterwards, they also did the analysis in another patient, in the patient 30. Here the problem is that despite having the more than 80% Tscm based CAR-T cells populations, they are not expressed within the patient even after 15 days. Afterwards, in this case they investigated the inhibitory cytokines levels, and they are able to see that the IL-10 levels goes up in the patient 30, whereas in all the patients it does not go up. This could be the reason why this CAR-T cells does not expand in the patient 30. And afterwards, after analyzing these two patients, now they wanted to check one more perspective or one more dimensions, that is whether there is a humoral response, that is anti-CAR-T cells are formed within the patient that is preventing these CAR-T cells to target the target cells. So since the CAR-T cells are made from the mouse construct, so they might be forming these antibodies, anti-FM63, which is basically the anti-CAR in this case, anti-CAR antibodies. So they check this anti-FM they check this anti-FMC3 antibodies in the serum of the patients, and they're able to find out that around 35% of the patients shows this anti-FMC3 antibodies formations or humoral response. And they also then correlated with this presence of B cells before the CAR-T cells infusions, and which is also indicates that when the B cells are present before the CAR-T cell infusions, there is more anti-FMC3 antibodies, indicating that these B cells plays a role towards these antibodies against CAR-T cells formations. So afterwards, to check this antibody response formations at different infusions, so they selected 4 different patients that have these B cells present before the CAR-T cell infusions, and then use after the first infusions, they are able to see that this anti-FMC3 antibodies formations goes down after some days, and in this case, after first infusion, they are able to see the suppressions of these tumor cells. However, when the second infusions are done in the same patient, they are able to see that there is a continuous increase in this anti-FMC3 antibodies, and in this case, in the later infusions, the tumor suppressions also not good to the extent they have seen in these first infusions. So this results in altogether indicates that whenever this CAR-T cells is used or the Tscm based CAR-T cells is used, these anti-inhibitory cytokines or these anti-humoral antibodies could be the potential therapeutic target to improve the efficacies of these CAR-T therapies. This is what they finally conclude from this paper. So I hope you understood the results that are presented in this figure and also the results presented in the previous figures about the paper. And if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.